Hello and welcome to Jack's Labs for ISCI 104. Today we're going to be doing the Great Cookie Dunking Lab, but first let's go over some vocab with the associated unit. There are a couple terms to go over, starting with the scientific method. The scientific method is the process of experimentations that is used to explore observations using the five senses and answer questions. Next are the steps to the scientific method. First, we're going to make an observation and ask a question. Then we're going to conduct research using trusted specific journals, online resources, conversations with friends, and books and articles. Next, we're going to form a hypothesis, test the hypothesis with an experiment, analyze the data and draw a conclusion, and then communicate the results so they can be found. Next is scientific variables. The first variable is the independent or manipulated variable, and this is the variable you change and is graphed on the x-axis. Next is the dependent variable, which is the responding variable. This is the variable you measure and is graphed on the y-axis. And finally are the control variables. These are variables that need to remain constant through the entire experiment for accurate results. Finally, regarding types of data, first is quantitative data, and this data is quantitative quantity, so it's numerical, and this can be graphed and can be countable, which is called discrete, or is measurable, which is called continuous. Next is qualitative data, and this is qualitative quality, so it is the descriptive and it is observational. So examples of that are color, smell, and texture. Now that we are done with the vocabulary, let's get into the lab. The question we are asking in this lab is how many seconds does it take to produce the ideal dunked cookie? The materials you'll need for this lab is the cookie of your choice. I will be using the original Oreo cookies, a gallon of milk or other liquid, a glass or a cup, a stopwatch, a measuring cup, and then a dish to place the cookie on when it's done being dunked. The independent variable is the amount of seconds the cookie is submerged in the milk or other liquid, and the dependent variable is the level of cookie sogginess, color, and consistency. The constants of the experiment is the type of cookie, amount of milk, type of milk, percentage of cookie submerged, room temperature, and temperature of milk. So what you want to do is do all these trials at the same time using the same materials. The control for the experiment is going to be a cookie that has 0 seconds submerged in milk or a cookie that was just taken out of the package. Now for the procedure. First, you're going to gather all the proper materials that I just listed. Next, we're going to get started. First, you're going to open the gallon of milk and pour the milk into the measuring cup until it reaches the 1.5 cup mark. If your measuring cup does not fill up to 1.5 cups, then make sure to add up the appropriate amount to get that full 1.5 cups. Next, you're going to take the measuring cup and pour the measured milk or other liquid into the glass or cup of your choice, and make sure that that glass or cup can hold that amount of liquid. Next, you're going to take the cookie out of the package and have your stopwatch ready to start the trials. For the trials, you're going to submerge the cookie in the middle of the glass and halfway down the middle of the cookie for however many seconds you start off with. In this case, it'll be 3 seconds. Then, after that 3 seconds is up of being submerged, you're going to remove the cookie and place it on the dish of your choice. Let it sit for maybe a second or two, and then start to examine and determine the level of sogginess on a scale of 1 to 10, and also describe any qualitative variables. Then we're going to get another trial in. We're going to remove another cookie and use the same milk and dunk it for the same amount of time, and then again describe the sogginess level. We're going to repeat those steps and replace the milk and cookies for 4 seconds, 5 seconds, 6 seconds, and 7 seconds. So then we have a lot of trials, and that should equal 10 trials, not counting the 0-second trials, which is the control. Once you've done that, you should have a subjective view of your favorite cookie sogginess based on the amount of seconds submerged in the milk. Thank you guys for doing this lab. I hope you enjoyed, and I'll see you in the next lab.